Bueno, hola a todos. Hello everyone. Welcome to the Young Investigator Symposium about cancer genomics and evolution. This is something that I organized this year and invited 3 great speakers, which will give us an overview of the current status of genomics and cancer evolution. My name is Luis Zapata. I'm working currently as an ICR fellow at the Institute of Cancer Research here in London. And I've been working in cancer for the past 6 years. Just a little bit of introduction to the topic for those of you that are undergrad students or just starting to look into what cancer means and what is the genomics behind this. So we know for a long time that cancer is a consequence of genetic alterations in the genome of cells. And more than this, it's also an evolutionary process that is defined by the accumulation of driver alterations, which leads to extreme proliferation or it leads to new phenotypes. Some of the phenotypes that you can observe in cancer cells are metastatic events, the capacity of invade other tissues, or the ability of resistance to drugs. We're going to see on some of these presentations today how, for example, cells can acquire their resistance to drugs. Another type of mutations that can lead to cancer is being known as methylation events, and we're also going to have one of our presenters today will also speak about this. It's also very important, the environment of the tumor, and how this environment creates heterogeneity, because not all cells are going to be able to survive on this environment. Some of them are going to be, some of these cells will have genotypes that are going to be advantageous, right? So this creates tumor heterogeneity. It creates positive selection, as I mentioned before, but it also creates negative selection. Okay, we're not going to talk about this today much about negative selection. We will focus on the topics that I just mentioned before. So cancer genomics has grown a lot on the last decade, thanks to the efforts of large consortia that have produced tons and tons of sequencing data, such as the International Cancer Genome Consortium or the Cancer Genome Atlas, right? This has allowed us, all scientists and data scientists working on the field, to uncover the landscape of genetic alterations on the cancer genome, such as short nucleotide polymorphisms or large insertions or deletions on such and others structural variation. So today, as I was mentioning before, we're going to see how this information has been exploited to understand tumorigenesis and ultimately how can we deal with this to find new treatments for cancer. So the lineup includes 3 great presenters. The first one is going to be Ahmed Acar. He's going to talk about developing quantitative experimental model system to study drug resistance. Our second speaker is Ignacia Fuentes. She's going to speak about how the tumor microenvironment or the cellular microenvironment impacts the development of some tumor types, in this case, the squamous cell carcinoma. And also we're going to see the identification of different type of genetic alteration. In this case, it's not genetic, it's an epigenetic alteration, how this can lead to cancer. So we can go to our first speaker, Ahmed. Ahmed was a colleague of mine at the ICR, where we worked together. He was working more on the experimental side, and I was working on the computational side. We are really good friends, and he's currently a group leader in Turkey, at the University of Turkey. Let's see, Ahmed, please.
Thank you, Liz. Thank you very much for the kind introduction. I will just start sharing my screen and let's double check if I do it properly. Can you all see my first slide of my presentation?
Yes, very good.
Okay, just a second because I want to put this. Okay, now everything's fine, right? You can see it properly and you can hear me. I assume it's yes. Just a second. Okay, thank you, Luis, once again. As Luis briefly mentioned, we work very closely. We are very good colleagues from Institute of Cancer Research in London. And I recently moved back to Turkey to establish my own research group. I am based in Ankara. So my research group's focus is mainly to understand how the drug resistance occurs, and in detail, whether we can develop quantitative experimental model systems to tackle or to control the drug resistance in our experimental setup. So by doing this, we leverage highly on the cancer evolution, how normal cells become cancerous, how distinct subclones can cooperate with each other to form the whole tumor mass. And by understanding this in the more detail, even a very close to the single cell level, using some cutting-edge single cell barcoding techniques, which I will present today, we are aiming to get close to understand drug resistance into more detail. So when I start my talk, I always go back to one of the old studies from 1976. This pioneering study by Peter Nowell especially showed us how the tumor evolution concept, the hypothesis of the tumor evolution concept is here, which we heavily use and rely on at the moment as well. So this beautiful picture, I think, as you also might remember, Luis also briefly introduced this picture, shows us how a single cell can acquire some mutations, and these mutations can give a higher fitness advantage to those single or a group of cells, which can then increase their proliferative capacity and create invasive and metastatic cascade to the second side of the tumor. And you may also notice in the top of this picture, there are some non-viable but antigenic cell types, which is a clear example for the other side of the picture, which is the negative selection. But what we are going to heavily focus on today in my presentation is the positive selection, how cells gain ability to grow more, proliferate more, with a higher fitness advantage, can be responsible for the drug resistance, which we see not only in the clinic, but on our experimental setups that we develop in the lab. So to continue, from 1976 until 2012, we haven't really seen much studies that focusing on the cancer evolution until this famous and very well-written review study by Malgreaves and Carlo Meli, which they visited Peter Nowell's hypothesis on the tumor evolution hypothesis, of course, in a much more detail. As you may also see clearly in this picture, we now have some bigger or smaller circles representing different subclones with the same genetic makeup. And when one of them is big, which shows clearly it has a bigger fitness advantage. And they can easily cooperate with each other, and from one tumor microenvironment to another tumor microenvironment with a distinct selective pressure, they may be either eradicated or they may be getting bigger to gain a better fitness advantage. And following by the treatment, which is represented by the TX here, you may select this resistant clone, which may be at the end responsible for the treatment resistance you are seeing in your system. Of course, these studies following 2010s, especially with the advent of the next generation sequencing, started to enter the research laboratories more and more. We start to see many more studies focusing on the cancer evolution. So one of the pioneering studies amongst these studies by Marco Gerlinger, in which he studied renal cell cancer, which he collected multiple biopsies from the one mass of the kidney, and he clearly showed their interaction within this multiple clones from the same tumor mass. So as you may have seen, the VHL is the clonal in here, represented with the blue in the phylogenetic tree. Ideally, one might think if you target this VHL, you may completely eradicate this tumor, which may be the strategy. But not always there is a available treatment options for every driver alteration that you are seeing in your system. But in most of the cases, you may be able to target one subclone in the one side of the phylogenetic tree, but the other side of the phylogenetic tree, the other subclone, may gain advantage to cover the rest of the tumor, which you may again at them see the failure of the treatment and the tumor growth in your system. So as you may have seen these snapshots of some papers, so applications of the next generation sequencing has become so popular, and people started to address many distinct mutational alterations or the copy number alterations in their systems in the pan-cancer studies. But one thing is obviously very clear in the clinic, especially. We now have a list of treatment options for nearly every cancer patient, but eventually we do observe the resistance to these tumors. So this is one of the famous examples in the melanoma case, vemurafenib. It's used in the clinic when there is a distinct alteration in the BRAF gene, V600E, and this specific drug has been shown to act and work nicely when it's being used initially. As you can see in the 15 weeks later after being treated. But as you can also see, following this treatment, unfortunately, these tumor mass comes back. So understanding the details of this, how this can occur, I think is very highly important. And this is the main interest in my laboratory in Turkey. As you can also see on the picture on the right, so there has been two main mechanisms has been proposed how this could be happening. So it could be directly due to either the presence of the pre-existing mutations in your system, and after applying a certain treatment, you may end up selecting these pre-existing clones, or through the course of your treatment schedule, you may start to generate new de novo alterations in your system. And obviously, it's not as simple as it's being pictured in this figure. So in most of the cases, it is being intermixed together, and it creates as a more complex puzzle picture. That's why it's still very hard to beat cancer in the clinic. So going forward, so this idea, especially, we are still not able to recapitulate the evolutionary dynamics in our experimental system, made us to think how we can closely mimic cancer evolution and ask a question, how we can study drug resistance in a quantitative way in the laboratory setting. To do this, we first realized the traditional way scientists, laboratories are studying drug resistance is not very much overlapping with the principles of the cancer evolution. So one of the main reasons for that, most of the drug resistance studies in the laboratory doesn't really mimic the realistic population sizes in the real tumors that we see. And another major point that we observed was in the laboratory old school, they use very low drug doses. And this is not very much realistic compared to the drug doses as being used in the clinic for the patients. And in most of the cases, people do a lot of replating, and they are interfering with the experimental system very, very often, and the length of their experiments in the laboratory context is very long. So these parameters are not often used or seen in the real patient in the clinic. Through this, you may end up seeing heavy bottlenecks that you are introducing into your system. And eventually, this may cause a genetic drift that you're observing in your system. And one of the most important parameters of the real tumors, which is the intratumor heterogeneity, you may end up losing that intratumor heterogeneity in your system. So based on this, we focused on understanding how we can develop a better experimental system which can mimic the real tumors in the clinical context. So to do this, we designed an experiment. We thought a lot about how to set up an experimental system. And we named this experimental evolution in the laboratory that we aim to set up the system. In this system, we use the hyperflask, which is way different than traditional way how people culture cancer cells in the laboratory. So this unique system allows you to plate 10 times more cells in comparison to the traditional way of culturing cells in the laboratory. So that was a way to get close to high population number in our system. And to be able to address these clonal changes, even at the single cell level, from that single cell level, the clones can be formed. So to address this, we use a cellular barcoding technique. So this is I'm going to explain this in the next slide in more detail. So with this technique, you can introduce a unique barcode into a single cell, and after your experiment is completed, by doing next generation sequencing, you can basically track back each of your clones in your system. And we perform these experiments in the non-small cell lung cancer cell line, and in this cell line, EGFR pathway is highly active, and to target this pathway and to generate the resistant cells in this pathway specific only, we use two inhibitors. So one of them was gefitinib, tyrosine kinase inhibitor targeting the EGFR, and another one was the trametinib, so which was targeting the MEK1 and MEK2, which is at the downstream of the EGFR pathway. So we performed these experiments. We treated 3 biological replicates of the initial population with gefitinib and 3 biological replicates of the initial population with trametinib. So these two act very differently. For example, for gefitinib, our experiment took 4 weeks from beginning till the end. When we hit with the high dose IC99 concentration of the gefitinib, we observed the population shrink, and then with the resistant clones started to emerge, we observed the population came back. And the same thing with the different drug, second drug, trametinib, acted different, and which took total 9 weeks. So you may recall we used this cellular barcoding technique, which allowed us to quantitatively measure our clonal dynamics in our system. So this lentiviral barcode system is 10 million complexity because of these 30 base pair DNA barcode, which is not present in the genome, is an artificial way of introducing into the genome in a stable way and labeling each individual cell with a unique barcode through lentiviral induction efficiency, bringing it down to very low. So by means of putting very low amount of virus, we were able to put a unique barcode into each cell. So after completing the drug resistance experiments, so this technology would allow us, if between the biological replicates, if we observe the same set of barcodes are enriched, so this would clearly indicate us this mechanism of resistance was pre-existing in our system. But if between the biological replicates, we observed a random barcode, selection of random barcodes, this would have clearly indicated us, so the mechanism of resistance in our system is because of de novo selection or combination of both, which I will show in the following slide. So we performed this experiment using the initial population, which has been barcoded. And in the first 3 biological replicates, we treated with gefitinib. And the second 3 biological replicates, we treated with the gefitinib. We treated with the trametinib, sorry. So you may recall, as I mentioned before, it represented with the blue in the first 3, for example. So these blue bars was the same barcodes clearly detected between the biological replicates. So this clearly indicated us that these mechanism of resistance under gefitinib in these, based on these barcode enrichment, was pre-existing in our system. We also observed same with the purple in the trametinib resistant one, but we also observed the double resistant group. The same barcodes was present in both drug treatment groups and which were pre-existing. And as you can see with the green, light green, we also observed some random barcodes selected, but they were different in each biological replicates. And that showed us these were de novo selection in our group. So therefore, we concluded this part is there is a polyclonal, because there is a pre-existing and de novo selection, there is a polyclonal drug resistance in our system. And by means of this quantitative measurement, because we were able to determine their frequencies, how many barcodes are there, and what is the percentage of these barcodes in the total population. So that allowed us to create a very quantitative system for each drug under this certain cell line through the course of selection under a drug gefitinib or trametinib. So we continue to understand more in depth molecular mechanism. So we asked the question, what might be the distinct genetic events under the gefitinib selection and under the trametinib selection. So before we do so this, we first confirmed our selected cell lines under gefitinib and trametinib were they genetically resistant. So we performed a very quick dose response curve experiments, and our 3 biological replicates under gefitinib were resistant to higher concentrations of the gefitinib in comparison to the DMSO and initial populations. We observed the same for the trametinib resistance. And when we performed the whole exome sequencing, we clearly observed under the gefitinib resistance groups, there is a MET amplification, but we haven't seen this MET amplification in the trametinib resistance. In parallel, in the trametinib resistance, we observed a CDK2NA loss in the system. In the same time, we haven't seen any CDK2NA loss under the gefitinib selection. So this clearly indicated us that under each drugs, there were distinct genetic events were taking place, and these may have been responsible for the resistance that we were seeing in our system. So before we further characterize and conclude how MET and CDK2NA is acting, we performed another very cool experiment. When I explain this, you will probably agree with me. So you may recall we performed the barcode sequencing at the end of our experiments, but these experiments took 4 weeks and 9 weeks, and every week we were changing the medium, and we were putting a fresh medium and putting a fresh drug into our system. We thought we shouldn't throw away the used medium every week, because we wanted to connect whether or not we can mimic a study, non-destructive, something like ctDNA from the blood people collect from the plasma, the DNA, and they can sequence it. Because we thought cells are dying under the drug, they are releasing their DNA into the medium every week, right? We try to generate resistant cells, and we are killing some sensitive cells, which were going to the medium. And in their DNA, there were pieces of the barcodes, right? It doesn't have to be intact and the whole DNA strand. Even if we were able to capture that barcode sequence from the used medium, we were able to draw a kind of conclusion, non-destructive, but time series evolution in our system. So that's why we thought this might be very cool. And which was the case. I mean, when we did the next generation sequencing of barcodes from every time point in 3 biological replicates of the gefitinib, we observed mainly 5, I cannot see it here clearly, but I think 4 or 5 distinct barcodes in an increasing frequency every week through the matching with the end of the experiment. Their frequency was increasing. That means cells are becoming resistant, and some of the resistant cells were still, going into the medium. And since the assay was so sensitive, we were able to catch a very low amount of DNA, very low amount of cells in the system to be able to sequence their barcodes. So you may, taking back the story a little bit back, you may recall MET and CDK2NA. In our gefitinib-resistant cell lines, we observed MET amplification, and trametinib-resistant clones, we observed CDK2NA loss. So we confirmed this with the DD PCR. So this DD PCR, the probe-based, we validated in our 3 biological replicates of gefitinib-treated lines. MET copy number is around double, maybe 2.5. And in the CDK2NA loss one, we observed the half loss of the CDK2NA. So observing this, one of the first things we thought whether or not there is a specific inhibitors for, let's say, for MET or for the CDK2NA is regulating another protein, which I will show you now. So we found there is a captamatinib, which is specifically acting on the this c-MET on the cell surface, and there is another inhibitor which is acting against CDK4 and 6. But CDK2NA loss negative, CDK2NA is negatively regulating CDK4 and 6. That's why CDK2NA loss might upregulate CDK4 and 6, and palbociclib against CDK4 and 6 might be a good candidate to try. So we checked this in our system, and we haven't seen any difference. So this was a pausing time for us. We start to think in depth more clearly, and we notice because of the high heterogeneity in our system, these drugs might not be active. So we thought we should sort, isolate individual unique single cells, and we should generate single cell derived cell lines and treat with the same inhibitors, whether or not distinct cell populations in the whole population might be more sensitive to these drugs or not. So that was the next thing we tried. So to do this, we use this single cell sorting system. So the difference of this single cell sorting system, it beautifully, with a camera attached it beautifully shows you a single cell has been sorted into your plate. So you have evidence you are really dealing with the single cell. Because one of the earliest and general problems with the FACS sorting system, you don't really see if you are sorting one cell or doublet. We really wanted to ensure we were sorting a single cell. And from that single cell, we generated a population of cells in a bigger plate. And we checked our inhibitors, whether or not they are working. Before doing so, we first validated different single cells in our system. As you may see here, not all of the single cells in our system has a high MET copy number. One of the clones showed nearly no MET high copy number in the system. But other 2 clones we picked in our system showed even more copy number gain than the average copy number status of the population. So we use these higher copy number single cell derived cell lines. We observed the same inhibitor which didn't work when whole population was tested. With a higher MET copy number, we observed these single cell derived cell lines were more sensitive to these drugs. So this clearly indicated that there is a high intratumor heterogeneity is ongoing in the system. So not all of the single cells are acting in the same way to be sensitive to the treatment that you are applying in your system. So we did the same thing for the trametinib resistance group. As you may have noticed, the single cell derived cell line clones that we studied in our system didn't show less than half of the CDK2NA loss in our system. So we checked this anyway with the palbociclib, and we haven't seen any sensitivity against these clones in our system. So this hasn't stopped us, and we pursued this angle further with high throughput screening approach, because we thought this CDK2NA and CDK4 and 6 is acting in the cell cycle. So that's very complex environment, and many proteins are highly regulating each other. So we thought if we screen these single cell derived clones in the high throughput screening with around 450 small molecule inhibitor library, we thought we may be able to capture some candidates, another cell cycle related inhibitor, and we may be able to combine palbociclib and this new candidate to be able to see if we can eradicate this population or not. So we performed this high throughput screening approach, and not surprisingly, our top candidates were all cell cycle related inhibitor. So this part of the study will still continue to be able to combine this palbociclib with these potential candidates to be able to generate more sensitive approach in our system. So far I presented the genetic changes in our system because I always focused on the barcode has been introduced into DNA, and under the drug we are observing the selection. But we also wanted to see phenotypic heterogeneity in our system. And to be able to do this, we performed a single-cell RNA-seq experiment using the same gefitinib-resistant and trametinib-resistant cell lines. And this is just a brief history how single-cell RNA-seq approach came to this point, starting from 2013 with the manual setups, and now is very highly high throughput system with the help of 10x Genomics. So you may recall in our system, we had a initial population pot, and then we had a trametinib-resistant cell line, and we had a gefitinib-resistant cell line. This is just a basic TSNE plot, and in this TSNE plot, as you can see, pot is in nearly one group, and the trametinib-resistant group is also represented with one group. And in the gefitinib-resistant groups, we observed two distinct plots in our system. Going into more detail, in this gefitinib-resistant group, in one of the gefitinib-resistant groups, we validated and we confirmed there is a MET upregulation in the system, as you can see here with the shades of the green in that MET amplified group, while the rest of the groups didn't show any MET upregulation. And when we looked at the trametinib-resistant group, only we observed specifically in that group CDK2NA downregulation, while the rest of the groups didn't show any change in the CDK2NA expression. Interestingly, one of the second groups in the gefitinib resistant, we clearly observed EMT phenotype. As you can see, the EpCAM expression is epithelial marker is downregulated, while the mesenchymal marker, the vimentin, is highly upregulated. So this EMT phenotype has also been studied in the past as a way to be responsible for the resistance, drug resistance that you may observe in your system. So to summarize, what I presented you today is that we aim to establish an evolutionary informed experimental model system. And in this model system, we were able to capture the ongoing intratumor heterogeneity. And by means of using this cellular barcoding technique, in our system, we observed polyclonal pre-existing drug resistance. And with the very cool setup that we were able to track evolution temporal way in a non-destructive way by means of collecting medium every week and checking the barcode frequencies from that used medium in the experimental evolution system. In our system, as we have seen, there is a polyclonal and the pre-existing drug resistance. So these multiple resistance mechanisms, also with the presence of the intratumor heterogeneity, might actually hamper to be able to see a clear collateral sensitivity when your system is bulk. But when you investigate it into a single cell level, you may be able to see some of the clones emerge from that single cell, might actually give you a collateral sensitivity, and through this inhibitor that you try, you may be able to overcome the resistance that you are seeing in your system. And with the power of single cell RNA sequencing technology, you can easily identify the ongoing phenotypic heterogeneity in your system. As I mentioned at the beginning, I worked 4 years at the ICR in London, and the work I presented you today has been done at Andrea Sotriva's group at the ICR. And I am a young PI in Turkey, and we just established our group. And now we continue to ask the next question based on the same technology, how we can establish collateral sensitive platform and the experimental evolution platform using different cancer cell line model systems in our lab. So my lab is currently heavily funded by the TÜBİTAK, which is the higher scientific council unit in Turkey. And I think that's all for today. Thank you for your attendance, and I'm happy to take questions.
Thank you, Ahmed. It's important for all the people to put questions in the chat. And we will take the questions for the end of the 3 of the presentations. I have a couple of questions for you, Ahmed. But I'll wrote them down. I wrote them down, so I will ask you later.
Okay.
It was a great presentation. Thank you very much.
I also would like to emphasize, I forgot to mention, sorry, we have an ongoing special issue collection about cancer evolution, and if you are interested in submitting papers, please feel free.
Yes, it's important to know. I also have one, and it's always good to get good papers on the pipeline. So now we will switch to Ignacia Fuentes. Ignacia did her PhD in the Max Planck in Germany. She studied biochemistry in Chile, and now she works as a director of research in the DEBRA Foundation. And also she's an associate professor at the Universidad del Desarrollo in Chile. So she will talk today with us about she will explain this. So please go ahead, Ignacia.
Yes, I think you can hear me, right?
Excellent. Excellent. Thank you very much, Ignacia. That was a really interesting talk. The same as before, I think we're gonna leave the questions for the end. So we're gonna have we have a couple of questions in the chat, and I have also a couple of questions. I guess Martin and Ahmed will also have something there. We'll move to our last speaker, so then we can go to the discussion and we can go to the question section. Martin Schaefer, he did his postdoc at the CRG in Barcelona, where we met. And then we started to work together, and we were interested on detecting signals of selection in cancer. And one of my most precious work has been with him as a last author. So he moved after that, after Barcelona, he moved to be a group leader at the Institute of Oncology in Milan, at the EIO in Italy. And he's gonna talk today about his latest research in cancer. Please, Martin, go ahead. Great, that was really interesting, Martin. Thank you very much. I think we are, can I share my slides so I can make like a little summary? and then take the questions. So this has been really nice because, as I explained before at the beginning, right, the idea was to understand a little bit more of cancer evolution and genomics, and I think the three speakers have shown an incredible work associated to these topics, right? I'm going to share my screen very briefly so we can just like recapitulate what we have like seen today, and then we will go to the questions. Can you see my slides?
Yeah.
Yeah. So from the first talk with Ahmed, right, he showed us how a cell, kind of like a technology to track evolution over time, it could inform the appearance or the emergence of resistant clones and to certain drugs. He showed different resistant mechanisms that were acquired during evolution, tumor evolution, and specifically he could also demonstrate the heterogeneity of this by performing single cell analysis. With Ignacia, he showed us the importance of the microenvironment and how other factors that might not be associated to specifically the cancer cell influence the aggressiveness and particularly some other properties that we see in tumors. With Martin, and we could also see that like not just driver genes, they're not just like point mutations associated to the establishments of tumors and the initiation of tumors, right? There might be other events that we have forgotten to look at into detail over the last years that might be associated to tumorigenesis. So I hope like with these kind of like very brief summaries for the audience, they can come up with more questions. We have some questions already on the chat, and we're gonna go over those first. Let me just stop sharing quickly. Okay, we can go first for like Ahmed. Ahmed, the question was, in general, what percentage of clones become resistant when you perform your experiments?
So for the gefitinib line, for the same cell line, for the gefitinib line, the existing population was roughly around, I can't remember on top of my head, but the frequency was around 20 to 15 to 20%, but I need to check the graph again. So that was the pre-existing population, which was further selected under the drug. But we also had a population which was less than that, and which was responsible for the de novo selection in this cell line under the gefitinib. And these percentages were different. These frequencies were different for the second drug, for the trametinib. And if you would perform the same experiment in a different setup, let's say in a colorectal cancer cell line, I would imagine we would discover different percentages, different frequencies of the pre-existing or de novo. So de novo is clearly linked, I guess, the length of your experiment and the drug pressure that you're applying. So again, if you perform this experiment, let's say in IC50, IC75 rather than IC95, we tried, you would have expect different degree of selection in your setup.
Yeah. Okay. That comes to one of the questions that I wanted to ask you. Like, what do you think are the pre-existing conditions that will favor de novo resistance or acquired resistance? Is there any kind of thing that you can screen, to know?
You are asking the relationship between the pre-existing clones harboring these pre-existing mutations with the new de novo alterations occurring, yeah. Right? The relationship. That's a very good point. It needs to be studied. To quick answer, so we don't know yet. So if your degree of pre-existing mutations are very high, whether or not you will end up new related de novo mutations, that's something we need to study, I think. But again, going forward with the single cell studies, if one can establish these single cell derived clones, then you will be able to, I think, address these questions much better way.
Yeah, I guess you can like mix you can mix clones, right? At the beginning, have multiple clones mixed or just one clone, and then you
That's experiment to do, I think. Yeah, you're very right.
Another question from the audience for you, Ahmed, is it says which mutation is associated to resistant to gefitinib in the EMT phenotype? Could you find any mutation specific associated in the EMT, or is just the EMT phenotype that confers the resistance?
The audience also remembers clearly. I think this is a good question. So in the gefitinib resistant one, we had two separated groups. So one had a clearly MET amplification, but the other one had the EMT, MET upregulation in the gene expression. So we did not have MET copy number gain in that particular EMT clone, as far as I recall. But there were, of course, different ongoing mutations, but I can't remember on top of my head if there was a specific driver alterations in the EMT clade, EMT group.
Okay.
But that's the interesting question as well. Maybe we need to revisit the data, I think.
Ignacio, Martin, if you have some questions, I have one more for Ahmed. Yeah.
Well, if I can ask one to Ahmed, because I was wondering, so maybe you mentioned something about this, but so how does heterogeneity in your system compare to true tumor heterogeneity? Like just degree. I mean, if you quantify the number of whatever drivers.
Yeah, you mean the system we use, the cell line, the degree of heterogeneity compared to a patient?
Yeah.
Yeah, I mean, of course, still that's a cell line. Although it's been, more purified, I would say, than the real tumor. We need to compare. I think the degree depends on the tumor type we are comparing, right? So in the same context, in the lung tumor, it will be a good idea to compare, I think, how many distinct genetic events they are observing in the lung tumor versus our system. But always, I think the cell line system will be more simpler, I think.
Of course it will, but you can model this, right? I mean, you modulate more than model. I mean, you can spike in as many clones as you want, right?
Absolutely. That's why the cell line model system are there for. You can easily modify any gene you want. You can start for overexpression. Absolutely.
You can have knockdowns of like the MSH3 and 6 genes, which are doing the tumor mutation burden. I mean, the mutation.
One thing is if it's aneuploid cell line, then you will have multiple copies. Then yeah, for those studies, people tend to use more haploid cell line, or diploid. Then they tend to be less cancerous. I mean, I would say. But again, if you introduce rather than knockout, if you introduce a kind of oncogene with a high copy number into a haploid or diploid line, then you can really observe the steps of the tumorigenesis, I think, in your system.
What do you think? Do you think like copy numbers are more associated to giving resistance in terms, or like increased expression? Could be methylation events as well, like Martin is seeing, right? Like that could be associated to drug resistance as well, no?
Yeah, absolutely. Certain growth, the certain receptors, especially in the EGFR pathway or MET, other well-known one as well. When they're in a higher copy number, they are directly reflected in the expression, and they trigger the signaling pathway, right? But also in relation to drug resistance, the famous example is for the EGFR, this famous T790M change or in the BRAF V600 change. One amino acid change can also, alter the pathway, or the bypass the pathway, and can cause resistance. I think it's in either way, but clearly copy number gain activates the gene expression way a lot, I think, than the point mutations.
Okay.
Can I ask him something, please? So I think it's really cool, the assay that you show. But it's huge. I mean, the amount of cells and plates that you use is amazing. How close or far are you from using cells from patients? I mean, could you simulate or mimic what you're?
Yeah, I mean, that's another project that we started in the lab. I mean, again, the basis is coming from my experience in the ICR in London, especially through our collaborator, the next lab, Nicola Valeri's lab in London. Now they're in Imperial. So the technology called patient-derived organoids, right? So that's the way forward, I think. So which we started here in Turkey as well, establishing the first colorectal cancer organoid biobanks. So this system is the most closely related system which can recapitulate the real tumor disease. So these are 3D culture systems, and they can maintain their gene expression, they can maintain their mutational profile, copy number profile, and everything in comparison to the patient. So that's the real most closely system that we may study, which we are planning to. So one of the ideas would be maybe introducing these barcodes into this organoid system and mimic these resistance experiments in the patient samples by developing organoids. That's one of the ideas actually we want to try.
And do you think, I mean, if the timing will allow to do the experiments first and then decide whether to treat or not the patient, or that's
that's the dream, right? So I don't know how close we can get to that. The idea, the dream is, at least in my mind, I don't know what the others are thinking about, taking a tumor from the patient and bringing it to the lab and establishing their organoids and giving the drug in the lab to the organoid and see how it's behaving, right? Rather than testing this in the patients. Because there are some approved drugs which is okay to try in the patient, but there are some experimental drugs as well, right? You may want to maybe try in the lab, but which you cannot try in the human. So that's why these platforms, this experimental evolution platform, which might be in the cell line, but there are limitations, but more relevant in the patient samples by establishing organoid technology, will be the way forward to check drugs in your list and do genomic profiling, all sort of, the experimental setup.
Yeah, we need to move to the next questions for Ignacia. Is there any association between the EV genotype and the frequency aggressiveness of SCC?
Yes, it's related to, I mean, there are different type of EV, and depending, for example, like patients with keratin mutations, they don't very rarely get SCCs. It's mainly related to the lack or the misfold of collagen 7. So that protein in particular gives, there are other types too, but mainly it's the absence of collagen 7 that gives this aggressiveness.
Yeah. So, okay, that's a good point, because it brings to my question, which is basically we tend to think that the cancer cells are acquiring, traits to become aggressive or invasive, or, driver high proliferation, We think that the properties are in the genome of the cancer cells, right? But it seems that in this case, is the lack of something in the microenvironment that already allows the cancer cells to invade, right?
Exactly. Collagen 7, it's not inside the keratinocytes. It's outside, it's in the dermis, it's below the basement membrane. So it's a protein within the dermis, the connective tissue. And that's the one that it's somehow driving other things like fibrosis, TGF-beta, other things. Yeah, exactly. So, I mean, I think it's a mixture of both. It's not just environment. It's
no, I mean, yeah, of course, it's a mixture of both, but specifically for the capacity of invasion, and that kind of brings me kind of like, can you separate patients into, categories of like low levels of lack of collagen 7 and good collagen 7, like boys that have a little bit, but and then compare the prognosis of these patients. Is it the fact that you have totally, can you make a gradient, totally disrupt collagen 7? Nothing. And this is very aggressive compared to the other.
I think it's something you can do. The main limitation is the number of patients. So it's a rare disease, so it's not very easy to get that. But yes, I mean, within the amount of collagen is usually related to the phenotype of the patient. So the patient is very, very severe, has a great lack of collagen 7 in between. But there's another interesting group that is even less abundant, so very more frequent, more rare, that is called the junctional one, that has problem with laminin. And those patients have a lot of wounds, like as severe as these ones, and they die very early, but they don't get as much SECs as the others.
Okay.
So they have this very chronic wound, all the environment, and they don't get SECs, or not as much. So those are a group that also is very interesting to compare. Like, what are the differences there? Because they do have collagen 7. Those patients do have. So that's not the impaired there. But again, the number of patients is what's limiting the studies here. And that's why there's a couple of groups in the world where everybody's sending their samples to, which they're able to do the research I showed you.
So then there's another question here. The question is, a los pacientes, the patients that you do follow up of the wounds, are they comparable in terms of the treatment? I'm not sure if it means you apply the same treatment to them, or like they were already with some certain treatment. I'm not sure.
So yes and no. So the disease doesn't have a cure.
Yeah.
So currently there's palliative care. So what you try, or the clinicians try to do, is that the wounds heal as early as possible, that they don't get infected and they don't have as much pain. So they are often trying to close the wounds, but sometimes they get infected, or they have systemic antibiotics given. So in general, they're similarly treated, but there's no treatment.
Yeah. Okay. Yeah. No, I see you point. What do you think is the role of the immune system in the process of the transition to squamous cell carcinoma? No, like to the SCC. Is it? Do you think it's related? It's something that we have discussed before, but like.
Yeah, I think it is. I think it's the immune dysregulation definitely has a point there. It's difficult to study, and that's why we were trying. The first panel that we used from these cells isolated from dressings was different immune cells like CD4, CD8, CD45. What was there? Neutrophils, and so there's a main opinion that so the wounds are open, and what the body first tried to do is like inflammate cells from inflammation getting there to somehow not get infected, but then you have the immune cells have to leave, and then the cells from the surrounding wound have to close the wound, right? But if the immune cells stay there because it's open, they cannot heal. So there is too much immune cells involved at some point. So the system is impaired, the neutrophil stays, and there's like hyperinflammation, and that's why the wounds are not closing. So there's something there that could be done as well. It's a multi-complex disease
process.
Yeah.
Guys, you have questions? Otherwise, move to the questions to Martin.
Just a very quick one. Did I understand correctly? So this ApoBec signature, this is specific to the squamous cell. You don't have it in melanoma, or not that frequently.
In melanoma, I'm not sure. So in the same SEC, so the type of squamous, the type of cancer is squamous that is usually appears in the skin.
Yeah.
Those ones are driven by UV mutations. So no ApoBec driven mutations.
But didn't you show in the end that you find the signature, this ApoBec signature?
Yeah. So yes. So in these patients, instead of having mainly the UV signatures, they have the ApoBec signatures.
Mm-hmm.
And that signature, it's also seen in another squamous cell carcinoma, the ones that people have in the head and neck. So the oral area that is not related to UV exposure.
Mm-hmm.
And that's because this enzyme is upregulated and giving mutations.
Is this related? This is my question now then. Is this related to a strong involvement in the microenvironment? Is this a stronger modulatory effect, or is this random?
I don't know. I don't. That's a great question. I don't know. I don't know. And that's a different. It's something to really take into account, because often clinicians, when they see a patient with a SCC, a skin SCC, they were going to treat them with other SCC, and the drugs that you give to patients with SCC in the skin, they're based on UV mutations, so a type of mutations driven by UV. So now they're considering using other type of drugs that actually tackle that kind of pathway. You're right. I have no clue where it comes from, but
Yeah. We need to go. We just have 4 minutes only of transmission. Martin, one question from the audience was saying, like, you have a method that with the correction you can better identify genes and pathways involved in specific cancers. That's basically, you know, like okay. And something also that maybe you can comment very quickly. How important you think that the interpatient heterogeneity affects the calculation of the covariates when you are mixing? Are they more stable? Are epigenetic covariates more stable between patients, or you know?
So of course this is an issue. Of course, I mean we detect better covariates and epigenetic changes with a low degree of heterogeneity, so which are you know present in a high fraction of all patients. I didn't explain this. In the beginning, I showed. Well, no one will remember. Of course, I say now, like 20 minutes ago, I showed you. But we had this little. I showed this little diamonds in this plots and how much variation we explained with the simple multiple linear models, and this was the consensus. In the consensus, we explain always better. So just the average degree of change, because of course heterogeneity is an issue. It's hard to model and it's hard to predict anything that occurs with it.
Yeah, you think this more is heterogeneity in terms of point mutations? It's, you know, is it more or less than compared to the methylation?
I think my feeling is less.
Okay.
So we have a higher degree of higher frequency in average of alterations. So there's a little bit less of heterogeneity within tumors, but also in the population. We find usually like I mean, we didn't systematically check within the same tumor but just like my feeling is that the similarity between patients is higher. That's why my assumption is, I don't know if this is actually valid, but that also probably like the intra-tumor heterogeneity is slightly lower.
Perhaps interpatient it's more similar, but intra-organs, you know, inter-organ it's more different, right? Because
this I don't know. I mean, so this is a good point. So I should have not answered so quickly. I don't know what's because we never checked. So I was just so we just checked. We compare between patients, but we never checked within intra-tumor.
Yeah, and I don't know if you have time for one last, very quickly, perhaps. It's like, so can you show that like the effect of APC or the tumor suppressor that are differentially methylated in cancer are not in normal cells? And with this, we finish and we close the question.
So yeah, we look at the change. So so what I showed on the y-axis usually is like basically the difference between healthy and tumor. So they are not already. So it's not that we look at the absolute levels; we look at the relative levels. So they're not already silenced
in cells. Okay, I think with that we've closed the session. Thank you very much to Ahmed, Martin, and Ignacia for presenting today. That was really good. I'm really happy to have you here today for the ALAG, and please, all the audience, the young investigators, can join the Genhoven Facebook, which is the sponsoring these talks. in this symposium. And thank you very much for participating, guys.
Thank you for the invitation.
Really great.
Thank you, guys.
Next time in real life.
Next time, yeah, next time in real life, yes.
That's for sure.
Next ALAG.